What if cancer cells could hijack healthy tissue not just by sending out chemical signals, but by literally donating parts of themselves? Imagine the very engines of cellular energy, mitochondria, being physically transferred from aggressive cancer cells into neighboring fibroblasts, rewriting their destiny and turning them into cancer's willing accomplices. But here's where it gets controversial. While experts have long focused on stromal and immune cells donating mitochondria to tumor cells, few have stopped to ask, could cancer cells themselves send mitochondria to non-cancerous cells and fundamentally alter the local microenvironment? Let's clarify the science powering this headline-catching discovery. Mitochondria, ideas familiar to high school biology as the powerhouses of the cell, do much more than just supply ATP. They also orchestrate cell signaling, stress responses, and decisions about survival or death. Once independent bacteria, these organelles have adapted to move between cells, a process dubbed mitochondrial transfer. Such cell-to-cell mitochondrial handoffs have already shown promise in healing sick tissues or boosting regeneration. In cancer, these transfers create metabolic diversity, helping tumors resist therapies and adapt to hostile hosts. Yet, until now, the story was largely one way. Stromal cells supplying mitochondria to malignant cells, helping tumors evade stress and proliferate. But this is the part most people miss. This study flips the script. It shows for the first time that cancer cells can send their mitochondria into normal fibroblasts within the tumor environment, reprogramming these cells into cancer-associated fibroblasts, CAFs, that help the tumor grow and invade. Let's unpack exactly how this process unfolds. First, researchers observed elongated thin tube-like bridges, TNTs, tunneling nanotubes, forming physical connections between cancer cells and fibroblasts. These TNTs, composed mainly of actin, allow mitochondria to travel directly between cells. Importantly, when fibroblasts were separated in transwell experiments, preventing TNT formation, no transfer occurred. Even more provocatively, inhibitors that block gap junctions actually enhanced TNT-mediated mitochondrial transfer, putting TNTs center stage. Advanced imaging and molecular techniques confirmed that mitochondrial transfer wasn't an artifact of dye leakage, which can be a confounding factor in cell biology. Species-specific genetic analysis showed that mitochondria from human cancer cells ended up inside mouse fibroblasts, a truly astounding cross-species handoff. Next up, does this mitochondrial transfer actually change the recipient fibroblasts? The data say yes. Fibroblasts that absorbed cancer-derived mitochondria exhibited marked increases in genes associated with inflammation, immune response, metabolism, and stress pathways, the classic traits of CAFs. These aren't just statistical blips either. Indicators of cell proliferation and production of extracellular matrix were all ramped up, well beyond the effects of mere exposure to cancer cell secretions. Here's a technical note for curious minds. Researchers used mitoception and fluid FM precision nanomanipulation technologies to transplant purified mitochondria directly into fibroblasts, confirming that the effect is due to the actual organelles, not just the process or secreted factors. Furthermore, the type and quantity of donor mitochondria mattered. Mitochondria from aggressive cancer cells induced stronger CAF reprogramming than those from non-malignant cells, and the transformation was dose-dependent, but only up to a point, suggesting a threshold effect. One of the most controversial findings, when fibroblasts receive mitochondria with impaired function, depleted mitochondrial DNA or chemically uncoupled, they fail to adopt CAF traits and their tumor-promoting power vanishes. This directly ties the quality of the donated mitochondria to the ability of a tumor to grow and expand. So what drives all this mitochondrial trafficking? The spotlight falls on MIRO2, a mitochondrial Rho GTPase protein vital for controlling mitochondria movement within cells. MIRO2 is highly expressed in actively growing, invasive cancer cells and at the leading edge of tumors. When researchers silenced MIRO2, mitochondrial transfer through TNTs plummeted and cancer cells lost much of their ability to stimulate calf differentiation and form tumors in mice. Conversely, overexpressing MIRO2 boosted transfer rates, but only to a certain level, beyond which fibroblast transformation plateaued. It seems there is a biological ceiling to this process. And this is the part that invites debate. Could MIRO2 become a clinical target for blocking cancer's manipulation of its microenvironment? Some scholars argue this is a promising direction, especially in notoriously stromal-heavy cancers like pancreatic carcinoma. Others caution that MIRO2 also supports basic functions in non-tumor cells, and disrupting it might cause unintended consequences. To wrap it up with a big-picture view, this study introduces a paradigm shift in our understanding of tumor-stroma interactions. Cancer cells can actively reprogram their neighborhood by handing over mitochondria to fibroblasts, inducing them to become CAFs and accelerating malignancy. The process depends critically on the presence of functionally intact, cancer-specific mitochondria and is regulated by MIRO2, a possible future drug target. Are these findings a game-changer for cancer therapy, or could targeting mitochondrial transfer inadvertently open new risks? What other diseases or regenerative settings might exploit similar mechanisms? Join the discussion below and share your perspective. Do you agree this could fundamentally change the fight against cancer? Or do you see unforeseen challenges ahead?